The goal of the following experiment is to conditionally induce epithelial cell transformation using an inducible expression system in zebrafish larvae. This is achieved by injecting the gene construct into a double transgenic zygote in the single-cell stage to induce expression in the superficial skin cell layer. Next, 4-hydroxytamoxifen is used to drive nuclear translocation of Cal-TA4 ERT2 and thereby overexpression of EGFP-HRAS G12V. Embryos expressing EGFP-HRAS G12V in keratinocytes are then prepared for live imaging by embedding them in agarose. Live imaging results show the interaction of innate immune cells with EGFP-HRAS G12V expressing superficial keratinocytes.
The main advantage of this technique over existing methods like the classical Gal4 UAS or Laux systems is that expression can be activated and deactivated upon addition or removal of 4-hydrotamoxifen.
Using this method now, we have the unique opportunity to understand some really fundamental questions in the field of cancer biology. Namely, how do cells evolve from a preneoplastic to a neoplastic state? And what is the role of the innate immune system in the earliest stages of cancer progression?
Generally, individuals new to this method might struggle because injection into single-cell stage embryos requires some practice.
The first step is to prepare an agarose injection plate to hold the fertilized eggs during injection.